So, welcome back. Today we're going to be moving into some more biochemistry. We just finished up talking about the four major types of biological molecules, or macromolecules, which are lipids, proteins, carbohydrates, and nucleic acids. And we're going to continue on now and talking about nucleic acids specifically. That was the last thing we talked about. We're going to carry on from there. So we did mention briefly DNA and RNA. We know that DNA stands for deoxyribonucleic acid. It's like a blueprint for an organism, and it's written in a code. This code has 4 letters, and these letters are A, C, T, and G. It stands for adenine, cytosine, thymine, and guanine. DNA is a polymer. We learned about this last time, where the polymer is DNA or RNA in other cases, and the monomers of this are nucleotides. Now, this is going to get into we have something called the central dogma of biology. This concerns the flow of information in a cell. It's somewhat limited, but it's a good place to start off with. So it says that all information starts out as DNA, right? And then it is converted into RNA. DNA is very long-term, is long-lived. RNA is a very short-lived copy. Just like if you wanted to save a phone number or something, you would take a picture of it, let's say. That is not the same. So in the same way that when you take a picture of something, it doesn't physically recreate that thing inside of your phone. It's just it's a recreation of it. So the same thing applies here. DNA is never converted into RNA. It is that RNA is a copy of DNA that is made using DNA as a template. And this process is known as transcription. That is, to transcribe is to take information from one place and put it in another. In this case, it is taking it from the form of DNA and turning it into RNA. It's from the same language it's from the language of genetics into the language of genetics. So there's no change in language. So it's just transcription. Like if you were to transcribe an interview, you might record an interview that you conduct, and then you would write it down later. From RNA, it is then translated into proteins. Now remember, protein is a 20-letter language as opposed to DNA and RNA, which are 4-letter languages. So in order to go from this language to this language, that has to be translated, and that is in fact what this process is called. It's called translation. So just in the same way that, like, if you were to try and go from Cyrillic to English or any Latin language, you wouldn't be able to just all those letters. And Cyrillic is a different number of letters. It's not like any Cyrillic letter is just like one of them is just an E and it's just written funny. It is an entirely different language. And this can give us a somewhat incomplete idea of how cells work, of how life works. It sort of implies that everything is predicated upon the DNA, and there's nothing, everything about what an organism is and does is based on DNA. And this is something that biology teachers have also pushed for some time, probably because we wanted students to take what we're teaching seriously. So we try and hype it up and say, like, no, this is super important. Everything about you is due to your DNA. And, like, that's not strictly true. There's plenty of stuff that is not due to your DNA. So these 4 letters, A, C, T, and G, again, they are adenine, guanine, cytosine, and thymine, A, G, C, T. We can classify these into 2 different shapes. Adenine and guanine are purines. Cytosine and thymines are pyrimidines. We can remember this because, or the mnemonic to remember this that my wife gave me is that I'm not going to write this whole thing, but agriculture is pure. So that's, oh my goodness, what happened there? Agriculture is purine. And then pyrimidines are the other ones. I don't have a fun mnemonic for that. And these always pair up in a predictable manner within the DNA, within a strand of DNA. A's are always opposite T's, adenines are always opposite thymines, and cytosines are always opposite guanines. So here we have, if we had two strands of DNA, if we had an A on one side, on the complementary strand, that would be a thymine. And if we had a cytosine on one end, we'd have a guanine on the other. And just the opposite, if we had a thymine here, we would have an adenine here. And these are connected to each other through hydrogen bonds. So hydrogen bonds keep coming back. They're all very important. The nucleotide structure, we talked about this, and that we have ribose versus deoxyribose. Again, we have our purines and our pyrimidines. We can see the chemical structure here. This is a 5-ring sugar. This is a pentose sugar. It is a 5-ring and also 5-carbon sugar. So 1, 2, 3, 4, 5. Right? You remember your organic. It's always a nucleotide is always made up of 3 things: a base, a sugar, a sugar, and a phosphate group. Now, here we have our sugar. It's down here. We have our nitrogenous base here, and we have our phosphate group. And phosphates, a phosphate group is just this phosphorus that's surrounded by 4 oxygen atoms. One of them is double bonded, but that's not terribly important. And here we have a more fleshed out chemical structure of each of the different nucleotides. Now, the language which all information needed to create an organism, not everything about it, but everything that's needed to create it, is written in those 4 letters. We can also say that DNA has an antiparallel double helical structure. So we have potentially two new words there. Double helical is pretty easy. Double helical just means if you follow here, we have this chain, and then it goes behind here. What the heck? My mouse keeps doing this. And then it goes behind there. And then we have a different chain that goes, we have a second one here. It goes behind, and then it goes in front. Maybe I should do this other one in a different color. We'll do this in red, maybe. All right, that goes in front. And here it goes behind. So double helical just means, so a helix is just one spirally thing, right? A helix is just like that, but imagine it 3 dimensions. So like a spiral staircase is a helix. A double helix just means there's 2 helices next to each other. And so that's pretty easy. The next word that we have to learn is antiparallel. Now we know that parallel lines, imagine that I can draw properly, right? Imagine these are parallel lines. Antiparallel does not mean like this, right? With a right angle there, let's say. These are perpendicular lines. Antiparallel lines are still parallel. It's just that now we're concerned with the directionality. Antiparallel lines are parallel lines that go in opposite directions. So that tells us something very critical, which is that these things have directionality to them. That these nucleotides have a direction to them. This is because they're not symmetrical, right? So if you imagine a metro car or a metro train, going in DC or whatever, those are symmetrical, right? There's an engine on either end, and all the cabins, all the coach, the cars in the middle are all pretty symmetrical. So it can, once it gets to one end of the track, it can just go the other way. This is not true of a car, right? When you look at a car is a very defined front part and a back part. There is a forward direction of travel. This is because cars aren't symmetrical, or we built cars to not be symmetrical, so we can tell which way they're going. That's not important for trains. So how do we get directionality out of these? Well, we can't say left to right or up and down, because those terms have very little meaning for when you're at the atomic scale. So instead what we do is we talk about DNA going from 5' to 3'. Now in organic chemistry, we always label our carbons because we're nerds like that. So we have 1, 2, 3, 4, and 5. This is a 5-carbon sugar. DNA is always written from 5' to 3'. So we have 5' So we can trace a line down through the DNA from 5' to 4' to 3', and then it's going to go and we'll put the next nucleotide, right? We'll put the next one, comes in down here, and we'll put our nitrogenous base there. That is going to connect up here, and then we could follow down that chain. We'll have our OH here, right? And we could follow this down again, and we could trace a line all the way down through an entire nucleotide chain and through an entire DNA chain from 5' to 3'. So DNA is typically written 5' to 3', but you can write it either way. You just have to mark which end is which. So this tells us that the fact that DNA is anti-parallel tells us that it has directionality to it. Each strand of DNA is also held together by hydrogen bonds. So you see in between here, this is where the hydrogen bonds are. This is what holds the DNA together. Right? See, these two strands never actually touch each other. They just sort of spin around each other. And between that space between them is connected, or there are hydrogen bonds exist there. So these hydrogen bonds exist between the nitrogenous bases. Again, the bases always pair up in a predictable manner. We call this complementary base pairing. In addition, if you look at DNA a lot of the time, if you look at pictures of DNA, you might see that it's sort of drawn very prettily like this, and it's very sort of symmetrical. This is not actually how DNA looks. DNA actually is offset here, in that we have a major groove and a minor, or we have, because they're offset, we actually have space here that is a major groove and a minor groove. So you can see the space here is smaller than the space between these two. So because of the, and some proteins will bind in the major groove, others will bind primarily the minor groove. And DNA is always right-handed, again, right? So if you imagine curling, you can curl your hand around this, right? You can sort of place your thumb up, and then your fingers would curl around that way. That's right-handed. DNA, so we're going to talk about this in much greater detail later, but generally, like, DNA is very stable, long-lived. Actually, we've talked about this in a fair amount of detail, in that the differences between DNA and RNA actually occur because RNA has this has a free hydroxyl here that is just not used in bonding, because when the DNA is all polymerized, these nucleotides, this OH is just an oxygen that's then connected to the next phosphorus. So we don't have any. Right? If we look here, we can see we have our phosphate group, and it's connected to two separate ribose sugars. So there's no hydroxyls that are just exposed to the air. So the existence of that free OH group on RNA is actually the only chemical difference between the two, and it's the cause of what the differences are. And I think I showed this last time. Here we have DNA. DNA is showing us DNA versus RNA. So RNA is a short-term copy of DNA. And that is going to be it for today. You're going to have a quiz as usual. You can find this on Canvas just below this in the modules. Have a great day.